Hello everyone, my name is Gaurav Kumar and in this video we will discuss about the types of microbiological growth media. Let's start. The topics covered in this video will be the classification mainly, the basis of classification and what are the types of media on that particular classification, simple media, selective media, etc. So let's start. First discuss what are culture media. When you are growing a microorganism outside their host species, you need to provide them the nutrition for their normal growth and maintenance. Culture media is basically a culture media contains actually that particular nutrients which are required for their normal growth and maintenance. When we are growing them artificially in lab, the microorganisms which are introduced in culture medium are called inoculum, and that microbes that grow and multiply in a culture are referred to as culture. Actually, the composition of the media, culture media is versatile, but the main factors on which it depends are on the screen: water, energy source, carbon, growth factors, minerals, salts, etc. We will be discussing them in detail later. Actually, a solidifying agent agar is used in many media as a solidifying agent. So why agar is preferred? Actually, there are substitute of agar, but they are not used, such as guar ample, silica gel. You know that guar is actually 50 times cheaper than the agar, but still agar is preferred because there are two reasons. The first one is it is not degraded by the most bacteria, and the second is it is inert. It does not participate in any reaction. Actually, agar has no nutritional value for the bacteria. It is produced by marine alga and it is easy to handle at the room temperature. It liquefies at 100 degrees Celsius and it solidifies at 40 degrees Celsius, which is comparatively easy to handle in a microbiological lab. So let's discuss classification first on the basis of composition. The first one is complex media. So the complex media is one whose whose chemical composition is not known. Actually, you are thinking, then why we use complex media if their chemical composition is not known? Because these unknown components are very good source of carbon, nitrogen, energy, etc. The unknown components are like peptone, yeast extract, meat extract, etc. And they are when we are growing an unknown microorganism, we don't know about their nutritional requirements, so we generally put these unknown materials such as peptones, etc., that fulfill their nutritional requirement. The composition is uh, the composition of the unknown material is dependent on two factors, mainly the source of attraction. For example, the peptones derived from cow or goat have different chemical composition and the physical process gone during the process to make the extract. Peptones are generally rich source of protein powder, and they are in the powder form. So the process undergone during the process to make extracts is also important, and it might affect their composition, chemical composition. The media is used for the cultivation of heterotrophic animals, and actually the main source is in this media are proteins which provide the which fulfill the need for the carbon, energy, sulfur, nitrogen, etc. The examples include nutrient agar, tryptic soy agar, tryptic soy broth, blood agar, conky media, etc. The second one is synthetic media. It is also known as chemically defined media. It is entirely free from the animal derived components. These components, the animal derived components, are generally unknown in their chemical composition, and that's why they are chemically defined, because they are free from the unknown chemical composition components. It is also used for the growth of autotrophic bacteria. Since these media are absent in the unknown composition, we have to provide them externally to fulfill the need of carbon, sulfur, nitrogen, and sulfur, phosphorus, etc. Their chemical composition is exactly known, and actually any media can be made uh, chemically defined by adding the known amount of the not amount but the known amount of the chemical composition materials whose chemical composition is known in the medium. Let's classify the media on the ingredients. First one is simple media, also known as basal media or general purpose media. These are these don't contain any specific ingredients. These are very general, supports the growth of all microorganisms. They don't contain any specific material, a specific nutrition or component which favors or alters the growth of any particular microorganism. They support the growth of all microorganisms. They are generally used for the non-fastidious bacteria to grow, and there is always a scope of addition of some nutrients rich in. Actually, there is always a scope of adding components which are good source of nutrition, such as blood, egg yolk, etc. These medias are used for the demonstration where a more prolonged survival of the culture is needed at ambient temperature. The second one is selective media. It favors the growth of a particular microorganism and suppress the growth of the undesired microorganism. For example, McConkey media, it is used for the specific growth of the entobac- Entobacteriaceae family, because since they are habitual in living the intestinal tract of the mammals, so they are adapted to the bile salts conditions, but the other undesired microorganisms are not habituated to the bile salt conditions. Actually, McConkey media contains a fairly good amount of bile salts, which suppress the growth of the undesired microorganism. Other media are also there, such as Lowenstein-Jensen media. This media is made selective by adding malachite green. It is actually a dye. Malachite green is a dye used as a also used as a dye in silk, leather, paper industry, etc., and also used as a inhibitor and pH regulator in Lowenstein-Jensen media. It suppresses the growth of the undesired microorganisms. Lowenstein-Jensen media is very much highly recommended for the growth of the Mycobacterium tuberculosis. In general purpose, they are used for Mycobacterium species. And let's look a picture at how E. coli looks on McConkey media and Klebsiella pneumonia on McConkey agar. Here is Lowenstein-Jensen media. The left one you can see in the diagram A, there is a slight yellow color which shows positive result. Yellow color is due to the colonies of the Mycobacterium developed. On the right side, more clear picture. Yellow colonies are of the Mycobacterium tuberculosis species. Let's go into detail about McConkey media. It is highly recommended for the isolation of Escherichia coli and other coliforms from pharmaceutical products. Let's take a look at the composition of the agar. To note that 3 points to note here is the ingredients include neutral red, crystal violet, and sodium chloride and bile salts. So, how McConkey media works? Actually, lactose monohydrate is a fermentable source of carbohydrate. Sodium chloride, as always used, maintains the osmotic balance of the medium. Pancreatic digest of gelatin and other nutrients provide vitamin and mineral source the growth of microorganism, and the non-lactose fermenters grow colorless, such as Salmonella and Shigella, whereas the lactose fermenters such as E. coli grow as pink to red color species. So where does the color come from? Actually, when the E. coli grow, it converts lactose into acid. The pH falls down, and the neutral red dye, which is already present, it is a pH indicator. It changes its color when the pH falls below 6.8, and so the different colonies, different color are observed for E. coli, who makes the pH fall below 6.8, and the other non-lactose fermenters which does not ferment lactose and the pH is not below 6.8. Let's discuss about differential media. Actually, it supports the growth of all media, but makes us to differentiate. It makes us to differentiate the colonies made on the petri plate. Actually, either the colonies are of different color, or maybe there are many factors by which we can tentatively uh, uh, differentiate between the different microorganisms grown on the plate, such as McConkey is also a differential media because the lactose fermenters produce pink color, whereas the non-lactose fermenters does not produce any color. The second differential media is the blood agar plate, and these are very important because they are extensively used to check the pathogenicity of the bacteria. And how you can check the pathogenicity is by studying their hemolytic behavior. Generally, the more pathogenic bacteria is beta hemolytic, means they produce pale yellow color colonies, whereas gamma hemolysis the produce no color around their colonies. Alpha hemolysis produce green color, and this this hemolytic behavior is extensively studied for Streptococcus and Staphylococcus species to check their pathogenicity, and we generally use blood agar. So let's take a look at how they looks like on the blood agar. Beta hemolysis, Streptococcus pyogenes, alpha hemolysis, Escherichia coli, gamma hemolysis, Staphylococcus epidermidis, and there is another media called mannitol salt agar. Mannitol is a 6 carbon attached in a straight chain and replace all the hydrogen with OH group. You get the mannitol. So Staphylococcus, actually the mannitol salt agar media contains high amount, fairly very large amount of bile salt, 7.5%, and this actually inhibit the growth of all gram positive gram negative bacteria, but only allow the growth of Staphylococcus species because they are the only ones to survive at that high concentration of sodium chloride. So let's take a look at another good, very important media, seablood agar media. The main point to note in their composition is the defibrinated seablood, which is actually 70% approx. So, how this works? This is recommended for the cultivation of fastidious organism and studying hemolytic reactions. What are hemolysins? Hemolysins are exotoxins produced by bacteria that lyse red blood cells. The composition there is list. What is the characteristic feature of this media? So there are three types of hemolysis. Generally, that this seablood agar media is used for to study the hemolytic behavior. Blood agar supplemented with seablood is used to study the hemolytic reaction of organism. So there are three types of hemolytic reaction: alpha, beta, gamma. Alpha, beta, gamma are actually in vague term you can say that these are the degree of hemolysis, means what extent they break down or lyse the red blood cells. In beta hemolysis, it is complete breakdown, no intermediate product is formed, and so the hemoglobins are destroyed to a colorless compound. But in gamma hemolysis, there is no hemolysis, and no change in the medium is observed because there is no hemolysis going. In alpha hemolysis, it is the intermediate product between beta and gamma, and hemoglobins are break down to a intermediate product methemoglobin, which produce a greenish zone around the colony. The nutritional requirements of the microorganism are fulfilled by tryptophan, peptone, and yeast extract, which provide carbon, nitrogen, amino acids, and vitamins. Sodium chloride, as always, maintains the osmotic valence of the media. Enriched media are basically when simple media are enriched with either in the form of blood, serum, egg yolk, that makes them enriched media. This media is generally solid and provide nutrient and environment conditions to favor the growth of a particular microbe. This is used for the growth of the fastidious microorganism. Uh, Examples are blood agar, chocolate blood agar. Actually, the heated blood agar is called chocolate blood agar. Bacteria are classified as alpha, beta, gamma on hemolytic reactions. We have already discussed. Look at the some uh, media on the left one, egg yolk agar, and on the right one, chocolate agar. How they looks. Hemolysis on the blood agar. Uh, we have previously also seen that. Um, Beta hemolysis, alpha hemolysis, and this is how C blood agar looks like. And the next one is reducing media. Actually, anaerobes might be killed by the exposure to oxygen, so we need a special media for their growth in which there is no oxygen, and such as sodium, we add some components like sodium thioglycolate that chemically combined with the oxygen present and depleted all the oxygen in the culture media. These media are also heated before use to drive off the dissolved oxygen. Let's This media, Robertson cooked meat medium, is highly recommended for the Clostridium species. Addition of extra 10% salt in this media is also recommended to grow Staphylococcus aureus species. Staphylococcus aureus. The ingredients include beef heart. The list is given on the screen. It is also recommended. It is actually recommended by the BIS also. So thanks for watching this video. If you like the video, please subscribe to our channel, and if you like this video, please like my video. Thanks for watching this video. Thank you.